So we're coming now to the final session of our day today, and this final session is entitled Research to Restore Vision with Dr. Michel Cayouette. And it's really a major pleasure for me to introduce Dr. Cayouette. He's a colleague in science and research, and he's also a co-member of the Scientific Advisory Board on which we both sit, and have very difficult sessions where we're in rooms for hours at a time looking at grants. So that's how I know Michel, and it's really a pleasure to have him come here to St. John's to give this talk today. And Dr. Cayouette is a neurobiologist at the Institut Cliniques Recherches de Montreal. I hope I got that right. I should. I have a French name, but I can't speak much French. That's fine. And that's in Montreal, and he's the director of the cellular neurobiology unit in that research unit. And Michel studies how photoreceptors develop from stem cells, as I talked about earlier in the talk today this morning, and how the genes in those cells can be turned on and off to control those cells to become parts of the retina. So he will provide an overview for you today on his own research in fighting blindness research. And his focus is on stem cell research, which is his own area of expertise over his training period and his current work in Montreal. So without any further ado, it's my pleasure to introduce Dr. Michel Cayouette from Montreal. Thank you.
Thank you very much, Bob. Stephen, how am I going to follow up on this? What I have to say is nowhere near as exciting or interesting or funny. But I'll do my best. It's a really. I think I'm going to repeat myself. I said it last night, and Bob mentioned it a few times. It's really a pleasure for us scientists to be invited to come and talk to a group of people that are affected by the disease we're so passionately trying to find cures for. And when I was contacted by the Foundation Fighting Blindness to come talk to you, I think I replied within a few minutes to Laurie's email saying, yes, I would love to be part of this. Because really for me, it's an inspiration, it's a motivation. After this morning, I just want to go back to my lab and start working again and really try to push my students and postdoc to work faster. And really, to hear from you and to hear all the questions you had, I was going from one group to another. Don't worry, I'm going to talk about stem cells. I was going from one room to the other and trying to hear all the questions that you had, and I thought it was so fascinating to hear from the people who are affected by vision loss what is important to them and what really they want to hear about. So Bob mentioned it in the introduction. I'm at the Clinical Research Institute of Montreal. I'm also affiliated to McGill University and the University of Montreal. And my work focuses mostly on trying to uncover how stem cells generate the different cell types of the retina. And I will try to give you an overview today of research to restore vision. And I will mostly focus on my stuff towards the end of the session, but I will also talk about other research going on in Canada. And there are different avenues of research for vision loss, as you've heard today. Some of them are gene therapy, for example, will slow down or they will preserve the vision that you already have. But they're very promising, although very challenging as well. There's hundreds of genes, as you heard, affected in different conditions, and replacing every single one of them is a very daunting challenge. So an alternative to this would be to use research to restore vision, even to someone who is completely blind. The good news here is that it seems like for most conditions, even though you lose your photoreceptor cells, which are the cells that are detecting light, as you all know, in your retina, these cells detect the light and they transform this light into an electric signal. So they are like an integrator of your retina, and they transform this light into an electric signal to send it to the other cells of the retina, which are responsible really to send the electric signal to the brain, where it's interpreted and into images that you can see. Now, when you're affected by RP or Stargardt or AMD, what happens is your photoreceptors degenerate and they die. Unfortunately, the retina is, fortunately, the retina is part of the central nervous system, which means that neurons of the retina cannot regenerate. Just like other parts of the brain or the spinal cord, once you lose them, you cannot endogenously regenerate your retina. But the good news, as I was saying, is that other neurons in your retina appear to be relatively intact. And so they are still there. And these are the neurons that transmit the signal from the photoreceptor to the brain. So those neurons are sitting in your retina, and they're just waiting for something to happen. They're just waiting for a cell to transform light into an electric signal so that they can be excited and then transmit this information to your brain so you can see. So, of course, now the challenge then with stem cells is to try to replace these photoreceptors, right? We want to give back to your retina. The photoreceptor cells are so essential in transforming light into this electric impulse. Now, the retina, as I mentioned, in humans and in vertebrates and in higher vertebrate mammals, in general, doesn't regenerate, but there are organisms, and we were talking last night at dinner, that can regenerate their retina. The fish, for example, goldfish, zebrafish, are able to regenerate their retina. You're going to ask me, why am I talking about fish and you're all humans? Well, and I was asked this question last night, why fish, why mice? Well, fundamentally, a fish retina is very much like a human retina. It has photoreceptors and the neurons that transmit information to the brain. Now, these fish can regenerate their retina from a pool of stem cells. And these stem cells are able to proliferate and generate new photoreceptor cells throughout the life of the fish. If you remove the retina of a fish, it will take a few days or weeks, and then it will regenerate its retina. Wouldn't that be something if this could happen in humans? Now, of course, there's a lot of research going on in Canada. For example, Vince Tropepi in Toronto, Ted Addison in Alberta, and many others are trying to find ways to uncover why the fish is able to regenerate its retina and humans can't. So I think this is a very promising approach. Now, but before we can do this, if the retina cannot regenerate endogenously, we need to have an intervention. We need to do something and try to put back these cells in the retina. So, and this is when we talk about research to restore visions. And when you think about restoring vision, you think about two things: prosthetic and stem cells. So the first, I'm going to first talk about retinal prosthetic. Not much, because this is not my area of expertise, but I know a little bit about it. And what I can tell you is basically this prosthetic is just an electric substitute to the retina. And it's a sort of implant, an electric implant. That's the electronic implant that's integrated into your eye, either in front of your retina or behind it, and basically will act as photoreceptor cells. And as I said, photoreceptors detect light, transform it into electric impulse. This is what this electronic device will do. It will basically transform the light into an electric impulse. Now, I was asked earlier by eSight glasses. I think you've all probably heard about that. These glasses are simply there to amplify the signal that the vision that you already have left. Some of you are affected by RP. You still have some peripheral vision, for example, and they can sort of increase what is there to try to help you to see better. But this is not what I want to talk about right now. These are different type of device. Here what I'm talking about is prosthetic that will basically regenerate new visions to someone who's lost it. So the device acts as photoreceptor cells, and they can stimulate the endogenous cells of the retina and to restore vision. Now, the problem with these things, it's very early days for this work, but I think it's promising. But the problem with this is that the prosthetic and the electronic device don't send the normal code to the neurons of the retina. They send signals that sort of stimulate these cells, but they're not the normal signals that these cells receive in a normal, healthy retina. So there are ways around this, and actually we have one of the world leader in Canada studying ways around this. His name is Gautam Awatramani, and he's also funded by the Foundation Fighting Blindness. And I can tell you, we fund only the best research. And I really, truly believe this, because I see, and as Bob mentioned, we see the grants coming in at the FFB, and it's really inspiring. There's a lot of good stuff out there. And so what Gautam has been able to do with collaborators in the US is to basically use, and I don't want to use complicated terms here, but it's something called optogenetics. What this is doing is, let's just forget about optogenetics now, okay? Think about a way to transform the cells that you have in your retina, not the photoreceptors, those neurons that send the signal to the brain. Make them able to respond to light, okay? And you make these cells able to respond to light, just like a photoreceptor. Then what you do is you put glasses in front of the eye. When the light comes in, these glasses filter the light, and then it sends these pulses in a way that is identical, or as closely identical to an endogenous photoreceptor would send these signals to the neurons of the retina. And adding these glasses can stimulate the retinal cells in a way that is very similar to what you have normally. And the result is a dramatic improvement in the acuity that you can get from these prosthetic devices. And actually, of course, this research was done only on mice so far. But the mice were blind. They were models of retinal degeneration. They had no photoreceptors. They've applied this approach, and the difference that you can see with what the mice can see is absolutely amazing. I've seen the result, and there is a dramatic improvement in the vision of these mice. So now this research is moving into trials into primates first. And hopefully this is going to happen in the next few years. And then eventually what the goal is to try to do these clinical trials in humans. Now, let me move into stem cells. I know that stem cells cause a lot of hype in the general public. And I would like to give you a realistic but hopeful view of stem cells. It's very important to realize that we are not going to stick stem cells into your eyes in the next coming years. It's going to take some time, okay? But there is hope, and I think it's a very, very exciting time in vision research. The reason is that I think that the retina is probably going to be one of the first part of the central nervous system that will be targeted with stem cell therapy, because it's a prime, it's ideal for these kind of therapies. So what are stem cells? The stem cells are cells that can generate any cell of a tissue or body, and they are able to self-renew. So they're able to make a copy of themselves every time they divide. So when they divide, they make two daughter cells, right? So you have a mother that divides and make two daughter cells. One of those daughter maintain the stem cell identity. So that way you maintain this population of stem cells indefinitely for the life of the animal or the human. And these cells have this great capacity of being able to generate any cell of a tissue or body. Now, of course, this makes them very attractive because you could think of regenerating certain cell types of the retina, like the photoreceptors, for example. Now, there are different types of stem cells. And some are controversial. Some are very promising. I'm sure you've heard about embryonic stem cells, which are taken from a fetus. Now, of course, there are ethical issues around these cells. But there are also advantages because they're very easy to grow and they can generate any cell of the body, obviously, because they're taken from a very, very early stage embryo just a couple of days after fertilization. Now, these cells, because they have the capacity to generate any cell of the body, they're very interesting for many, many different conditions, not only vision loss. Now, there's also adult stem cells, and one of the greatest discoveries of the past 10, 15 years was made in Canada for adult stem cells in the eye, and it was made by Dr. Derek Vanderkooy's group in Toronto, and what they've discovered is that the eye in humans contains adult stem cells. So adult meaning that even in an adult, I have some, you have some, everybody here have stem cells in their eye. These cells are located, if you look at your neighbor and look into their eyes, you will see a ring, a very dark ring around the iris, this colored part of your eye. That's where the stem cells are located. And what they have shown is you can take these stem cells from a patient, donor, mostly so far it's been done only with dead patients who have donated their eye, and they took the cells from this area, grew them in a dish, and generated millions of photoreceptor cells in the dish for grafting. Now, this was amazing news, and it's a discovery that was made in Canada. And people are following up all around the world on this, and it's a lot. There's a lot of promise, obviously, but there's also a lot of difficulty with using these stem cells. For example, it's very hard to control their division rate. So you don't want these cells to divide forever. You don't want to have a tumor in your eye when you graft them. You want them to generate the right cell type. So I mentioned that these stem cells can generate any cell of the tissue. In this case, for example, the eye or the retinal stem cells can generate any cell of the retina, but you don't want them to generate all the cells of the retina when you have RP and all you care about are your rods. And so research really now is focused on trying to make these cells become the right cell type. In this case, for example, rod photoreceptors or cone photoreceptors, we don't want them to make the other cells of the retina. Now, of course, there's a limitation also with using stem cells in terms of immune rejections, for example. I mean, of course, if you take stem cells from a dead patient and grow them in culture and try to graft them into someone's eye, there could be immune reaction. But there's ways around this that people can use now. And there's also an important advance that was made recently called Induced Pluripotent stem cells. What are these? These are basically, this is just a trick that researchers found to transform your skin cell into a stem cell. So the potential here is to take, for example, a swipe in your mouth and collect a few cells on the skin of your mouth and grow them and add a few genes in them, and they will turn into stem cells, and they will be able to generate any cell of interest, in this case, for example, retinal cells. Now, the advantage of this is that you get around the immune rejection problem, because you could get from someone's a swipe from someone's mouth and then grow his or her cells in a dish and graft them back. The problem with this is, of course, you don't fix the genetic problem. If you have a mutation, it's in all of your cells in your body, so there will be a need for a combination of therapy here. So that's something to be aware of. Nonetheless, there's been a pretty amazing success with using stem cells to restore vision. A couple of examples. Advanced cell technologies, I'm sure you've probably heard about them. It made the news quite a bit. Have been able, in a recent clinical trial, in 2 patients only, it's a very small group of people, but still promising. One had macular degeneration, the other one had Stargardt disease. And what advanced cell technology has been able to do is take embryonic stem cell, in this case, and differentiate them into RPE cells, these cells that lie at the back of the eye that you heard about this morning, and graft them in the eye of these patients, and they had some vision recovery. This was exciting because it provides a proof of principle that, yes, maybe one day we can use stem cells to regenerate, to restore vision. But these were RPE cells. What about photoreceptors? So far, there's no clinical trial that I'm aware of, at least, that are using stem cells, photoreceptor derived from stem cells into grafting and going on in clinical trial. But there's a lot of research that's been going on in mice, and blind mice have received photoreceptor derived from stem cells into their eye and actually restore some vision in these animal models of retinal degeneration. So this is promising because it means that the proof of principle is there. You can take a stem cell, differentiate it into photoreceptor, graft it in the eye, and you restore some vision. But there's still a lot of work to do. So two problems with this is, first, the cells don't really make, the stem cells don't really make a lot of photoreceptors. I said that the stem cells have this great capacity of being able to generate any cell of the body or any cell of the retina, in the case of adult retinal stem cells. But they don't generate photoreceptors very efficiently because they generate all the other cells. So we need to be able to sort of narrow their capacity into generating only the cell we're interested in. And so this is one big focus of research, and I'll come back on that because this is stuff we do in my lab. The other problem when you graft the stem cells or the photoreceptor-derived stem cell from stem cells, they don't tend to integrate very well in the retina. They don't tend to spread very well everywhere. They tend to stay where you put them. So there's exciting research going on in Derek Vanderkooy's lab using hydrogel to try to spread the cells around and try to have as many cells as possible across your retina. The other thing that's going on that's very exciting is that we can use drugs to try to promote the survival of the grafted cell. Because the problem when you graft them, a lot of them will die. And you end up having just a fraction of the cells that you've grafted in the eye surviving. And so you want to increase this survival. And so by giving some drugs, people have been able now to improve this survival rate dramatically. And this also is some research going on in Canada and abroad. And now, the work that's going on in around the world that I was mentioning is trying to focus the stem cell into making photoreceptor cells. So there's been some really exciting work, I think, that came from a lab in the UK, the group of Dr. Robin Alley, who have discovered that actually stem cells change over time. They're not always the same. They seem to change their mood, if you want. At some point in their life, they seem to like to make photoreceptor cells. At other times in their life, they seem to prefer to make other types of cells of the retina. So he found that actually the time at which you graft these stem cells into the eye is absolutely critical in generating a high proportion of photoreceptor cells that will be functional. He found in mice, this is studies in mice, that there is a very narrow window of a few days where you graft the cells and you get a restoration of vision in these mice. Now, if you do the same experiment just a little bit earlier or a little bit later, you get absolutely no integration and no vision recovery. Now, this tells us that there is a very strong genetic program operating in the stem cells that help them to make photoreceptor cells at the right time. We need to uncover how stem cells decide that it's time to make the photoreceptor cells. Even more excitingly is that when he grafted, in a very recent study that was published just 6 or 9 months ago, he grafted these cells and now recorded from the cortex. Because someone could say, well, you graft these cells, yes, you can record from the retina some signal that says that these mice are seeing. But it doesn't really show that you get reconnection to the brain normally and that the cortex can interpret this signal. What he has shown now is by doing this cell grafting therapy, the mice are not only getting signal in their eye, but they're also interpreting the signals in their brain. So they're recording from the cortex. They put these mice in a visual test in a maze, and they were able to see and sort of find their way to the food where the food was placed. And all this kind of visual tests were used to show that actually these mice do see. It's not simply that they have some signal going on in their retina. Now, I think this is very exciting, and now what we're doing, and just to finish on this, is to try to understand how the stem cells make photoreceptors. I mentioned there is this narrow period of time that the stem cells are very good for to generate photoreceptor cells. So we've discovered about a year and a half ago a gene that is expressed in the stem cell at the time when they're making photoreceptor cells. This project was just funded by the Foundation Fighting Blindness last spring, so we're very excited about this. And I have a couple of news to tell you. So this gene is expressed in the stem cells, and it's able to control when the stem cells are going to make photoreceptors. If we inactivate this gene using genetics in mice, we can prevent the stem cells from making photoreceptor. Now you'll say, well, that's not so good. No, but what we can do is if we misexpress the genes, now we put it earlier at a time in the stem cell, very early, when they're not normally making photoreceptors. And all of a sudden they start making tons of photoreceptor cells. So what this is telling us is that this gene might be key to coax the stem cells into making photoreceptor cell in a much more efficient manner so that we can get way more photoreceptor cells from stem cells. And not only that, but what we found, in addition to this exciting result, I think, in stem cells, is that the gene continues to be expressed in photoreceptor cells. And it seems like this gene is there in adult retina in the photoreceptor cells very specifically. So this now is a gene that was not known to be expressed in photoreceptors. So this is a new gene that we've discovered expressed specifically in the photoreceptor cells. And we are now doing experiment to try to figure out what it's doing there and if it could be involved in maintaining the survival of the photoreceptors, for example, or helping them to function normally. And so this is research that's going on in my lab. We're really trying to use mouse genetic to tackle this problem. And eventually, our goal is really now to collaborate with people who know what they're doing in terms of human adult stem cells, like Derek Vanderkoy, for example, and try to convince them that maybe adding this gene in the stem cells will help differentiate the stem cells into many more photoreceptors that will be much more useful for therapy. Now, I just want to finish on another aspect of stem cell that is also very exciting. I mentioned that fish can regenerate their retina, and I said, wouldn't it be great if we could do this in humans? Well, it turns out that there is a lot of research going on now to try to reactivate the endogenous stem cells that you all have in your eyes. And there are two different populations of cells that are able to reactivate upon a certain stimuli. The adult stem cells that I mentioned earlier, and also a type of cell in the retina called the mula-glia. And so those cells are glial cells. But it seems like what they do is simply they provide support to your retina. They make sure that everything stays together. They provide some survival signals and maintain the retina functioning normally. But it turns out that what people found is that you can stimulate these mula-glia to proliferate, even in the adult retina after an injury. And when they still proliferate, they're now able to regenerate new neurons. And this is not grafting here. This is just stimulating these cells to proliferate with the drug. So I think, again, this is very early days, and I just don't want to tell you we're going to do this in your eyes in the coming 2 years. But I think this is the kind of research that is very promising and that you should be looking forward in the future. And this is also things that are happening in Canada and also abroad, obviously, because we have an international community of vision research. So I hope I gave you a general overview of stem cells that is realistic, but also hopeful. And I hope that you will see this as a motivation for you, as a something to look for. And I can guarantee you that all researchers across Canada and internationally are working countless hours in their lab to try to really improve stem cell therapy. And I think we will get there. It's just a question of time and money. And time, I can provide. Money, I try to provide too. I do make donations personally to the Foundation Fighting Blindness, and I strongly encourage you to do the same. If it wasn't for funding of the FFB, I couldn't have discovered this gene I just told you about that's active in the stem cells. So thank you very much for your attention, and I'll try to answer your questions. Don't be shy. I know that my students are always embarrassed to ask questions, and I will wait. You're like my students. Yes.
Thank you for a very interesting talk. Earlier we were speaking about the defective gene, and will a defective gene unwind the effect of stem cells so that you would find yourself having to repeat the stem cell cure?
I'm not sure I understand your question completely, but as I mentioned, if you take stem cells from a patient that's already affected by RP, for example, and this patient has a mutation, you take his or her stem cells, they are also affected by this mutation. So you would need to, cure that mutation, either using gene therapy, for example, before you could graft them back into this patient's eye. The alternative is to take, stem cells from a healthy patient, a healthy donor, and use anti-rejection drug therapy to try to prevent rejection from the patient. And I think this is probably more where things are going in trying to provide immune drug therapy to prevent rejection from the body and using the healthy stem cells from donors. So is it what you meant, or did I get this right? Is it answering your question? I'm not so sure.
So I think that what you have explained is the stem cell from the healthy donor will not be affected by the unhealthy genes that remain in the in the body. It is a complete localized cure. That's right.
Yes, and it's for most disease. There's always, of course, possible complications, especially with AMD and the blood vessels leaking and all this stuff. But still, you could restore at least for some time, and that would be great, right? But the other thing I should mention, and I wanted to mention something, I forgot. It doesn't matter.
Dr. Cayouette?
Yes.
I wonder if... just like this? Yes. I wondered if donations from relatives that didn't carry the mutated gene or the damaged gene, would they have a better chance of not being rejected by the patient than if they were from a stranger?
Yeah, it's a great question. You have a better question than my students. Are you interested in doing a PhD in No. Yes, there is an increased chance, but it still needs to be tested. You still need to see if this relative is immune compatible. It's just like when you want to do a graft for the leukemia, for example, and you need to take a graft from someone in the family. You will first look in the immediate family because there's more chance of finding someone that's compatible in the family, and then you go around. So that I would imagine it's the same for stem cells. Yes. Yes. There's a question in the back.
Yeah, well, I have AMD, and I'm well in my 60s now. My daughter is having a baby in February, and she says, "Mom, maybe I can take the stem cells and do." So, what do you do with the stem cells in the meanwhile, and where are they stored, and what's the cost of it, and is it how long?
Yeah. Okay. I don't want to tell you what to do. And I think it is a very personal decision that you have to make. I have colleagues who have kept stem cells from their baby. I have other colleagues who don't believe it's going to be useful. And I personally have mixed feelings about this. I think it could be useful. The problem is there's so few cells from the umbilical cord. I guess that's what you're referring to. That if are we going to be able to really use them in 60 years, or if there is a problem? There's debate around that. It could be useful for certain conditions, but certainly not for all. And so, unfortunately, I cannot give you a clear answer. I think it's just a decision that you have to make in your own belief. If you think, what I can tell you is there's a lot of stem cell research going on now, and certainly not only in vision, in all sorts of disease, and certainly knowledge and efficiency of differentiation of these stem cells and proliferation will increase over the years. So where are we going to be in 25 years from now? I cannot tell, but certainly promising that there will be improvement, and maybe it's worth keeping them. Oh, the storage. So this is, it goes in, and the cost, right. So I think this will vary from, there are different companies doing this. It's relatively expensive, but I don't know the cost, the exact cost. It's certainly several $1,000s. Where do they store them? They store them in liquid nitrogen at minus 196 degrees, which is very, very cold, colder than Newfoundland in winter. And they keep. Of course, there's a lot of confidentiality going on, so you need to, they make sure that your cells are kept in confidentiality, and so they're secure, they're safe somewhere in a bunker. Yes, in the back.
With respect to AMD, are you aware of any research looking into the possibility of using stem cell therapy to repair the leaky blood vessels themselves?
Yeah. I'm aware of people interested in that question. Whether they've made progress, that's another story. I think there is, well, Bob might be more aware than me about that, but I think there is some people in the U.S. interested in using stem cells to differentiate them into endothelial cells, which are the cells that make up the blood vessels. What I just don't see is how are they going to get them at the right place in the eye, in the retina, and so there is. It's very early days, I think, for this. It seems like stem cell graft is much more advanced in terms of RPE cells, obviously, because there's clinical trials in humans, and now more and more in photoreceptors. But for blood vessels, I'm not aware of anything that's going on that would be like dogma changing.
Yeah, it's fair, Michel, I think, and we had this question earlier in my session, and I think it's a great question, and what I can add to what I said earlier for the gentleman's question is that the other complication is that the blood vessels in the retina are probably very different from blood vessels that are in the rest of the body. So it's going to take, again, a lot more basic research, and there are individuals like, as Dr. Cayouette said, in the U.S., Maria Grant's lab down in Florida is studying this, and those blood vessels in the retina are extremely different from other blood vessels. So I think what you'll have to see first is a lot more basic research done in the lab to understand those blood vessels, to really then move to stem cell therapies for repairing the ones that are diseased in these diseases.
So we need to take baby steps before we can I think this is something I might not have mentioned enough. We need to take baby steps before we can get into clinical trial to get something out to the patients. And these baby steps are done in the lab with fish and with mice and with animal models and with cell culture because these are very complex questions and that need to be addressed before we can move into further ahead in the trials.